All right, guys, welcome back to Underground Science, and in this video, we'll be talking about Toll-like receptor 9. All right, so let's go and get started. So Toll-like receptor 9, again, like we've been talking about for Toll-like receptor 3, 7, and 8, is found inside the endosome of the cell. So not on the cell surface, but inside the cell, specifically inside the endosome of the cell. All right, so what type of cells can we find this in? Find Toll-like receptor 9 in? So that would be again similar to toll receptor 7 and 8, you can expect to find toll receptor 9 in cells like your B cells, your dendritic cells, and your, of course, your macrophages too, right? Macrophages. So, so let's go and see how toll receptor 9 gets activated and what that leads to. Okay, it's actually going to be pretty similar to toll receptors 7 and 8. Okay, so toll receptor 9 actually recognizes and gets activated by unmethylated, okay, unmethylated CpG DNA. Okay, so CpG DNA, right? Let's write this down. CpG DNA is basically Um, it's the part of your DNA that's rich in cytosine and guanine. Okay, so you can see this a lot in certain promoter regions. Okay, so promoters, certain regions of the promoters, right, of your DNA can be rich in cytosine and guanine, and that's what toll-like receptor 9 responds to, which would be unmethylated CpG DNA fragments. Okay, because usually it's important to know that usually CpG DNA is methylated, right, in mammalian cells, okay? But in certain in like bacteria or certain viruses, right, we can have unmethylated CpG DNA inside of them, which can help our immune system, right, of course, distinguish between like foreign DNA, right, so pathogens, or our own DNA. All right, so let's go and see how toll-like receptor 9 gets activated and what that leads to. So let's say we have a certain virus, okay, and this virus can actually be like, for example, HSV, right? Herpes simplex virus, or it can be certain things like cytomegalovirus, the cytomegalovirus, or Epstein-Barr virus, right? Which we'll talk about later. We'll talk about these viruses later when we talk about the micro in the micro playlist, okay? Because for example, the Epstein-Barr virus, right, is important when we're talking about things like Burkitt lymphoma. Okay, so that will be talked about in our white blood cell playlist, where we talk about certain white blood cell disorders, lymphomas, leukemias, and so on. But let's go ahead and see how these viruses. We're focusing on toll receptor 9, right? So let's go and see how toll-like receptor 9 gets activated due to these viruses. So if we take these examples, right, and just say that this is our virus, and our virus here has our CpG DNA, right, inside it, and it's not methylated, unmethylated, right? And we have a dendritic cell or a macrophage, right, here nearby that will see this virus and phagocytose this virus, okay? And we know when that happens, that leads to a phagosome production. Okay, that leads to the production of a phagosome or an endosome. All right. So what we're going to have now is a phagosome or an endosome here, like that. And then we'll have our virus here with our CpG DNA, right, inside of it. Now, for our endosomes, or for our toll-like receptor 9, right, molecules, which are present inside the endosome like this, for it to recognize for it to recognize the unmethylated CpG DNA, we need this covering to break down. So what happens when you form the endosome pH decreases, okay, which we discussed in last video too. So the endosome pH actually decreases. When that happens, you activate certain enzymes like proteases, which can help degrade the covering of the virus, right? Or the pathogen that we're talking about in this case, helps degrade the capsid or envelope or the covering of pathogen. So once you degrade the covering, right, now the CpG DNA can get exposed to these toll-like receptor 9 molecules. All right, so once you degrade this covering, right, we can activate these toll-like receptor 9 molecules. Okay, so once the toll-like receptor 9 molecules get activated, they dimerize. So you get two toll-like receptor 9 molecules that dimerize. So we can expect something like this, right? We'll draw the dimerization like this. This can be our toll-like receptor 9 molecules that have dimerized. Once these receptors dimerize, it gets activated, right? And then once we get activation, this can lead to the activation of the MyD88 signaling pathway, which we reviewed in the previous video when we were talking about toll-like receptor 7 and 8. And it actually leads to the same thing. So once MyD88 gets activated, then we get the activation of two main things, right? One would be activation of nuclear factor kappa B, which we know would then go to your nucleus, increase gene expression, lead to the production of many different cytokines like interleukin 1, interleukin 6, interleukin 8, interleukin 12, TNF alpha, and so on, right? That's not an alpha. All right, and so on. So that pathway increases the production of cytokines. The other pathway, right, once my D88 can get activates, right, that can lead to the activation of IRF7, just like we talked about in our toll chapter 7 and 8 video, right? And we know that once IRF7 gets activated, that can lead to the production of our interferons, like interferon alpha and interferon beta. Now, what's the use of interferons again? Right, remember interferons induce an antiviral state. Okay, that's really important because you want to contain the virus. You don't want it to spread to other cells nearby, whether that be through the production of protein kinase R, right, which we have reviewed in our previous toll-like receptor videos. It's important for, stopping the translation of certain viruses, right? Or whether that be through increasing expression of MHC1, so that can lead to your cytotoxic T cells, right? Your CD8 positive cells, CD8 positive T cells to come and destroy whatever cell that's infected with the virus. So you can contain the virus that way. All right. So, like, if all of this was happening inside a dendritic cell, for example, right, and it was responding to some virus, for example, like herpes simplex virus. So not only would we see the activation of our MyD88 pathway, which we know would lead to the production of many cytokines, right, through the activation of nuclear factor kappa B, but it would also lead to the production of interferons, right, through the activation of IRF7, okay? But what will also happen, since it's a dendritic cell, right, or a macrophage, those are antigen presenting cells. Right? So if we're talking about dendritic cell, for example, not only would it lead to the release of cytokine interferons, it would also present the viral particles, right? Of if we're talking about HSV in this case, it would present the viral particles to our CD8 T cells to activate them, right? Because we know these antigen presenting cells, like dendritic cells, for example, if it processes certain viral particles, right, viral proteins or tumor proteins, it can take these proteins or parts of the virus right, and present it on its MHC1 complex. So let's try to see how this works with an example, right? So let's say we have a HSV virus or our cytomegalovirus or Epstein-Barr virus, right? So if we have this virus here with its unmethylated CpG DNA, right? What happens is our dendritic cells or antigen presenting cells can take this up and present it at MHC1 because we're used to seeing our antigen presenting cells taking up viruses or pathogens, right? And presenting on MHC2. But that happens if your antigen presenting cells take in the pathogen and degrade it, right? And then they present it on the MHC2. But if the if our antigen presenting cells, right? So let's say this is an, oh, not the same color. Let's say this is an antigen presenting cell that we have here. And if this antigen presenting cell, right, phagocytoses this virus and takes it in, right, then we have our virus now. So let's say this leads to our antigen presenting cell like that, right? And then we have our virus inside. All right. So if this happens, what can happen is the viral antigens, right, stay in the cytoplasm. This is actually called cross-presentation, which we'll go over in more detail when we do our immunology playlist. But in cross-presentation, what happens is instead of the antigens going inside the lysosomes, right, of this antigen-presenting cell, the antigens can stay in the cytoplasm or the cytosol where we have proteasomes, right? Because remember, all nucleated cells have MHC1. So if we have a virus infecting a normal epithelial cell, right? A normal epithelial cell, same thing happens here, is that the virus can get degraded by proteasomes, right? So we have proteasomes inside our cytosol, and then that degraded viral particles or the viral peptides can get put on MHC1 and expressed on the cell surface, right? Or with our MHC1. So this is going to be our MHC1. Same thing happens in our antigen presenting cells. If we undergo cross-presentation, right, this virus, right, can get degraded by proteasomes in our cytosol of our antigen presenting cells, right, which can lead to many different viral peptides, right? Now these viral peptides can go into the endoplasmic reticulum, right? And get processed and put onto MHC1 molecules. All right, get put onto MHC1 molecules, which are inside our endoplasmic reticulum. And then from there, we can actually express that on the cell surface. So we're going to have our MHC1 here and our viral peptides here, right? And then, of course, we know who does MHC1 respond to or who does MHC1 attract, right? That would be our CD8 cells. So if we have a naive CD8 cell here, with its T cell receptor, right? And we, of course, we know that we have our B7 molecule on our antigen presenting cells, right? So our B7 molecule like this, and we have our CD28 molecule on our CD8 T cell, CD8 positive cell, right? So this is going to be our CD28. This will be our B7, right? So we know this interaction will then happen. Let's label this as our MHC1. And then we can therefore activate these CD8 cells into cytotoxic T cells, right? So it can get activated fully into cytotoxic T cells. And we know with the Th1 subtype, if we go back a little bit, our Th1 subtype of our CD4 cells can release interleukin 2. Remember, that was one of the cytokines that can release, and this can help further activate our CD8 cells and help proliferation, okay, induce proliferation of these cytotoxic T cells. All right. So if that happens now this cytotoxic T cell recognizes, right, this viral antigen right here, and then it can go on and kill infected cells, right? Infected cells nearby that are infected with this specific virus. Okay. So that's how we can basically activate CD8 cells through MHC1 when talking about antigen presenting cells, okay? Because we usually know that antigen presenting cells present antigens on MHC2, but if it undergoes something called cross-presentation, then it can take these viral particles and present on MHC1 like we showed here, which can further activate CD8 cells, and then those CD8 cells can then go to nearby cells that are infected with this specific virus. Okay, so that's the basic pathway for toll-like receptor 9. Okay, the main thing to know again is it responds to unmethylated CpG DNA, so islands of DNA where you have a lot of cytosine and guanine. Okay, that's not methylated. It's going to respond to that, and then lead to the release of interferons and cytokines, which will then help increase inflammation, vasodilation, the normal steps that we see with inflammation, right? And it's going to help attract white blood cells to the area. Interferons will help reduce the spread of the virus, and then that will, help contain the virus and help or lead to your immune system fighting off whatever pathogen that is infecting your cells. Okay, so that's the job. That's the basic overview of toll-like receptor 9. All right, and I hope this video helped a little bit. Hope this made a little bit of sense. Hope it wasn't too confusing. Thanks for watching, and see you guys in my future videos.